Maxime has a PhD at ISTEM, right? And Maxime is the co-founder and chief scientific officer of TreeFrog. So maybe you recognize yourself as a postdoc that was thinking about a startup creation some years ago, as we are discussing just before. So please, would be very happy to know more about your journey, and thank you for being here with us.
So hello everyone. I finished my PhD in 2011, so it was quite a few years ago. There is quite a journey to start a company and to push, especially in this case, a technology in the context of cell therapy. And what I would like to give everyone the impression of is that this is definitely something that you can do, and it's definitely something that you don't necessarily need to accept the advice that are going to be given to you. So you can take whatever I'm going to say with a big grain of salt. One of the things I think that everyone that is smart enough has is this imposter complex. You know, like, am I the right one? Am I doing good enough or everything? I would argue that in the context of biotech, what is really the most important is the science and the relevance of the technology that you are pushing forward. And you will have people that have an MBA, that have expertise, and in the end, if you want to start this journey, you will have to push yourself, and you will find people, and you must find people, but you will have to push yourself. That's very important. Can I get the... oh, yeah, it's there. Okay, cool. Okay, so just a little bit of details about TreeFrog. So we were founded in 2018, but the dirty secret is that I started to work on the idea right from the start when I arrived in my PhD in 2006. Actually, I met with my co-founder, Kevin Alessandri, who was working at Pierre et Marie Curie University in Paris. So I was also in PhD in ISTEM, in Évry. And we actually met as postdocs in Geneva. And we met in 2013. So there is a 5-year gap between us meeting and the starting of the company. And that's not, it did not just happen. It was actually because we had the chance to find support in the French ecosystem. And we've had a lot of support from the French ecosystem. That's very important, because when we were both in Geneva, when I was starting to say that I wanted to come back to France to start the company, I've had very cold returns, like, why coming to France? Are you crazy? And no, I'm not. And it's actually, the French ecosystem is actually a very good ecosystem to help push things that are quite far from the mark. In the US, you can have a lot of money on things that are already almost ready. To some extent, it's true also in Switzerland, but if you really have to push hard and do science, I would argue that France is best positioned. I mean, obviously I'm very biased, but nevertheless. And so actually what we did was we moved in 2015 in Bordeaux, and we were sort of undercover, writing grants, public grants, being helped by the CNRS, being helped by the university. Then we had iLab, and there is this whole ecosystem of funding that actually helps do these things. And we were very lucky to be very successful within that ecosystem to get money to push forward, and that was really instrumental. So again, we met in 2013, we came to France in 2015, and we really founded the company in 2018. So we were 2 at the inception, and now we are 160 actually. And very important, we actually brought in very early complementary skills. And here you can see it's listed biophysicists, stem cell biologists, and bioproduction engineer. That's actually true. It's not really a third, but that's not so far from that. But we also very early had people that were knowledgeable about starting a company. And we actually brought in people that were completing us. And it was, I would argue, a quite efficient strategy because we've been able to raise quite a lot of capital. So obviously it's in the pocket of no one because this is money to be spent on developing. Developing a cell therapy product is very expensive. And so basically we have a Parkinson's disease therapeutic, and the whole point was that our technology, and I will speak a little bit about it. I don't want to put too much time on the technology because I'm not here to sell my can of soup, but rather to give you some insight as to how this journey was done. But the idea with Parkinson's disease was actually to have a twist using physics to deal with biological issues. And typically, Parkinson's disease cell therapy is not a new thing. We did not invent anything there. Lots of people have been working on that field for a long time. There are companies that are now in clinics, TreeFrog Therapeutics being one of the chief ones. It's not the only one. What we do is we actually change something that is kind of important, which is that most people work with progenitors. So progenitors would be cells that are somewhat resilient to harsh treatment, very much more so than neurons. Neurons are basically the little princesses of cells and are very fragile, very efficient, but very fragile. And in our context, we actually engraft microtissues that contain mature neurons, and that's basically the only way that I can think of now to be able to engraft mature neurons in the brain that are produced with a cost-effective strategy. And we are also working on acute liver failure because our technology can really scale up massively, which is kind of moot in the context of Parkinson's, to be honest, but is very important in the context of acute liver failure. The very good thing that we managed to do after, if you count the years, 10 years, 11 years into the project was signing Vertex. So Vertex is developing a type 1 diabetes microtissue drug, and we have secured, so it's biobucks. We don't have this money right now, but we signed a 780 million deal with Vertex, which puts us in a good space, very important right now. And we've filed a lot of IP. And from the get-go, we already had 4 patents that we had developed in university when we started the company. And we've not stopped ever since. It's very costly, but that's what got us the deal. Okay, so then what exactly are we aiming to be? So we aim to be a technology leader. We aim to use, again, physics to deal with biology issues. And the idea is to think about what makes cell therapy hard to develop. And it's a lot to do with CMC, with manufacturing. Manufacturing a cell is, in a way, easy because the cells divide themselves. You don't have to really produce them. I mean, you do, but you don't have to fully produce them. But they are very hard to characterize, and they are very hard to scale also, because even though biologics have paved the way for bioreactor use in the context of producing drugs, it's not the same thing to produce a protein, an antibody, and a cell. Because the cell that you want to put in the body, you cannot afford to adapt it to the bioreactor. You cannot have mutations along the way that make them survive better, because then what would it mean in the body? Okay, and there is a lot, because it's a new way to develop things. We even have to develop surrounding technologies, and that's a lot of work. Okay, So the whole point of what we do is sort of summed up here. This is an epiblast-like structure. So these are iPSCs. So the first cells, not really the first cells, but let's say the last step that is truly multipotent, pluripotent, sorry. That's a bad one. Pluripotent in the body. So basically, all of the cells that you see here retain the capacity to generate any cell that is present in the adult body. And actually this shape, so this is a slice. You can think of it as the peel of an orange. You actually have a lumen inside. The apical part of the cells is facing the lumen, and the basal part of the cells is facing out of the object. And that's actually very reminiscent of the organization of the early days of the embryo. And we actually shape the cells like that, not to have to make a stand in science, but actually to have better production capacity. Because the cells, when they are in the conditions they're supposed to be, they perform better. So I'm probably not gonna, oh, and there's even a problem with the font. So good excuse to switch. Oh, again. Okay, no, well, very quickly, oh, sorry. Very quickly, that's kind of important. So I'm not gonna go through the details, actually. It's a very good excuse to go fast. It's not very interesting to you, but nevertheless, cell therapies have a lot of promise, but the problem is that it's very hard to scale. It's very hard to scale and maintain good reproducibility, and it's even harder to scale, maintain reproducibility, and not kill most of the cells when you deliver them. So these three things in a row, aligning these ducks is not easy. And our answer is basically to mimic what happens in vivo, trying to be as physiological as possible. And there's a little bit of a trick, and I'll show you after. The consequence of that is to aim for maximum efficiency and thus better access for patients, because then the costs can be driven down and the rationalization as to how to industrialize that can be really achieved. So basically, everything we do is linked to pluripotent stem cells. The pluripotent stem cells in culture are actually an artifact. In vivo, it's actually kind of a short time span. You don't get pluripotent stem cells for a long time. It's actually the beige window that you have here. So it's a few days, but in these few days, you actually accumulate about 10 divisions. When you think about it, any cell in your body, and it's not really true, it's not true for the gut, it's not true for, but don't quote me totally on that, but it's about, in average, 40 division from the first cell to the last cell. Again, exceptions, but in average it's that. So 10 divisions along this is a lot. It's basically the quarter of the risk you run of making a mistake while dividing the cell. So that's quite important, and that's a step in the body where you can invest massively, because basically the mother will be infinite in terms of energy as compared to the needs of such a small amount of cells. So that's a time in development that is under very high pressure to do good, because if you make a mistake at this step, the whole body is going to get the mistake. And so that's a very good cell to actually amplify, because the trade-offs for these cells are quality, 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 and speed. But quality first. And the thing is, by using this as a means to scale up cell therapy, being able to amplify massively these cells, if you respect their topology and the biological environment they're supposed to have, they're going to do a good job. Okay, so how do we do that? Actually, what we do, so it's a rendering on the left. So it's cut artificially, obviously. So what we do is we protect the cells, because the cells know how to self-organize, but they are not meant to resist a bioreactor. The physics of a bioreactor is very harsh, actually, and the more you scale, the harsher it is. So basically what we do is we put them in a comfy space. They have space to grow, so it's only a lining of alginate. Alginate is a very interesting hydrogel, because it's an hydrogel that can very quickly reticulate, and that you can shape, because it can be, again, as an hydrogel, a liquid first. So that's what you have on the right. Basically, we generate thousands of capsules per second, so it's slowed down 600 times here. And it allows us, each of these drops is actually what you have on the left. And it enables us to have up to 10,000 cells per drop, which means that the encapsulation step at which we have to put the cells, it's not to produce first these capsules and then we put the cells. We have to do that at the same time, so that's a hassle, but we do that at very high speed, so that's not actually limiting. So that's what's happening in one of these. So you see that there are some dead cells to begin with, but then it structures as a luminized epiblast-like structure, protected by the alginate. It grows in an extracellular matrix that we can feed in the encapsulation. It forms a cyst, and you will see that there is no accumulation of dead cells, because you would see them. You see the one in the beginning, and you see that during proliferation there is no more dead cells accumulating. And that changes everything, because it means that every cell that you don't lose is contributing. And you don't have selective pressure, because if cells don't die, having a mutation that makes you survive more is not all that advantageous. Okay, so we transferred that from a 30 ml bioreactor to a 10-liter bioreactor, and as you can see, the higher up you go in size, the better the performance, because that's what you should have. A big bioreactor is better controlled than a small bioreactor, and people tend to have poorer performance in bigger bioreactors only because of the physics of the bioreactor that starts to be harsh to the cells. So if you basically put a firewall of alginate that makes that they don't see the physics of the bioreactor, they actually behave better when going up in scale. We use the technology, so the technology is not a bioreactor. Technology is what you have in the center, so it's an encapsulation technology. We have a machine that we developed that enables us to go into any bioreactor, and then you can either use it to amplify iPSCs or embryonic stem cells, and or differentiate them. I think, maybe we'll see, no, we don't. And the alginates we did not invent, but it's very interesting because it's very easy to remove. Basically just have to remove calcium in the liquid, and then the alginate will go back in solution. You can wash it off, and it's fast enough. It's less than a minute, it's tens of seconds, fast enough that it's not unsettling to the cells. And we've used this technology to do central nervous system neural cells, liver cells, cardiac cells, epithelial, retinal pigmented epithelium, hematopoietic stem cells, and NK cells, all of that from iPSCs. And we've also tested culture of adult cells in it, and it also works very neatly. So, and I will just take only a few slides again, and I will stop so that you can ask me some questions. I think it's more interesting than me selling my soup. Okay, so we are working in the context of Parkinson's and acute liver failure and type 1 diabetes as a collaboration. Okay, so that's a good thing that we don't have the deal terms. I think it's not really interesting. And let's go very quickly with Parkinson's. Yeah, okay, problem with, my bad, I should have checked that it's okay, I can do this without, without what's written. So basically, why did we choose Parkinson's? Because Parkinson's is not a rare disease. It can be useful to go quick in clinics, but the whole idea of the technology is to be able to scale up, so it would be sort of self-defeating to target that. The mechanism of action in the context of Parkinson's is quite understood, which is that dopaminergic neurons, being the first one to die in the disease, fail to provide the proper amount of dopamine in the striatum, and basically it's very impacting on the life of the patients, and for a time, and only for a time, taking per os dopamine can lead to a very interesting solution. But it's a neurodegenerative disease, meaning that the disease continues to progress, and ultimately the patient cannot be dealt, the symptoms cannot be dealt with dopamine. And so basically the idea of putting back in the structure that is supposed to get the dopamine, the dopamine producing cells makes a lot of sense. And that's basically the strategy that has been developed for 30 years in the field. And the slight twist that we have is that we are able to produce more efficiently, more reproducibly, and we are able to produce the cell of the mechanism of action rather than the cell that's going to differentiate into the cell of mechanism of action. And I stop there, and I will take questions. If there are.
Thank you so much. Very interesting to see this overview. Do we have questions to Maxime?
Pardon. Ah oui, merci. Thank you so much for your insightful sharing. I would have two questions. The first one, which is not very technical, why TreeFrog? Why frog and why tree?
That's a good question. I'm going to be fast. That's a question I get a lot.
Maybe just to make the second one, which is much more technical, I was curious to know a little bit more about the encapsulation that you did, and did you try other hydrogels? Why alginate? And did you make some screening of what kind of environments are good and gels to the encapsulation?
Okay, so I'm going to answer the second first and then okay, so why alginate? Because alginate is easy to actually source under GMP. So produced in a fashion that is acceptable to use in human. The second thing is that it's kind of a marvel of biophysics, I would say, because basically it's you know, probably if you've seen a fjord in Finland or something, you see the water, but underwater you have kelp, you have like huge laminaria algae, and basically they have everything you want. It's tough because it's supposed to withstand the flow of water. You need tough because you need to protect from the bioreactor. It's the toughness is give or take what you have with cartilage. And the smaller an object, the less tough it has to be to withstand the bioreactor. So it's kind of small, it's like hundreds of microns, and it's tough as cartilage, so it's like tough enough. The second thing is that algae need light to go through, and that's very useful because you want to be able to see what you do, just to check if your bioreactor is going the right way, even to characterize and anything. It's also permeant to gas because the algae needs to feed from the CO2, from the water also, and it's permeable to quite big proteins too, but not too big. So the algae needs to protect themselves from viruses, and the alginate is doing that very fine. But everything up to an antibody, give or take, will move. So that's very interesting. That's why it was mostly used to format the cells to go in the body, because it can prevent antibody response and cell attack. But that's not the reason why we use it. We use it to shoehorn into the bioreactors. And we basically remove alginate before using it because we want integration with the body. And then, first question, why TreeFrog? Because we're French, and one joke was like, you know, we eat frogs. And it makes no one laugh. Only English people are getting the joke. Like, Japanese are like, what the fuck? And the US are like, but whatever. It was a self-deprecating joke. And the second thing was that amphibians are models of development, and clearly cell therapy hails from development. So that was that. And then the second thing is that why tree? Because it sounds nice. And also because trees are marvels of microfluidics. And the physics that we use to put the cells in the bioreactor is basically microfluidics. I mean, like putting all this water high above for use is amazing.
Another question?
Two quick questions. One, I was wondering if now that I mean you have a licensing deal, etc., are you facing pressures to move the company to the US, for instance, from investors or from partners? And maybe answer this, and I'll ask a second question.
Well, yes, obviously, but that's the whole point. We came to France because we wanted to do a company here and not in the US. So it was not to have a skipping stone, and like I don't remember how you say that in English, but So the idea is to stay in France, and I think there is a lot of relevance to that because there's very good research to be done here, and clearly the market, not gonna lie, we have a few leads. All of them are in North America, so you don't really have a choice. But the thing is, it doesn't mean you need to take your company and put it only in the US, because the costs in the US are tremendous. For example, the current facility that we have, and we are in an incubator, it costs us, I think the calculation was 200 times per meter square what it costs us to be in Bordeaux. So it's Bordeaux, but nevertheless. So, and also you hire very good people. We are in Boston, clearly, you have a very good ecosystem, but it costs a lot too. So yeah, there is a real rationale to balance the two. So we will not expand the company, but then you also need to have investors that follow that. And that's always the problem. The money is much easier to have in the US. And the typical US investor is going to be, So.
But will it be feasible for you to do the manufacturing in France and treatment in the US, or does the product have to be produced locally?
Apparently it's not impossible. Clearly not the preferred option for the FDA, but you can do that. And nevertheless, what I deep down don't want to jeopardize is that to be able to produce in France for Europe. That's not something we're gonna negotiate with, and maybe we will have right now. Vertex, it's their product. They will not come to France to produce it. Let's face it. But the idea is to have production in France for Europe. Ultimately, I think it makes sense, and it can be economically sensible to produce in France for the US. It's not impossible. It's not going to be easy. It's going to be tough, but why not? And there will be some level of US for US, Europe for Europe, Japan for Japan. And we all know how the world is going. So I think that's the most likely outcome.
And then I have just a very naive question on cell therapies. How do you deal with the immune response of the patients?
That's a huge question. You choose your indication. Basically, that's what we've done. So in the context of Parkinson, there is this very debated notion that the brain is an immune privileged space. It's not entirely true. It's not entirely false either. And basically, the level of tolerance you have to achieve to be able to stay in the brain is not the same. It's very known from a solid transplant. If you engraft a kidney or a liver or a heart, you don't need the same level of immune suppression. So clearly, there is a huge question about how to have tolerance long-term of therapeutic products, and that's why we have Parkinson on one hand, which is maybe the least hard to meet in terms of requirements. And on the other hand, we have ACLF, because ACLF, it's a lot of cells, but you only need them for 4 weeks. So that's, and then there are endeavors to make truly hypoimmune cells, and we are looking at that very closely, but that's not, today no one can claim that they have a truly definitive solution for this question.
Thank you very much, Maxime. It was really nice for us to have you, and a real pleasure. And